I'm interested in questions related to DNA content, and a sort of hallmark of cancer is that many cancers or malignancies have either too much or too little DNA, an abnormal DNA content. And this is true for a lot of different types of tumors, but in particular it's very true for acute myeloid leukemia. I'm really interested in two questions. First, what are the effects of either having too much or too little DNA? And then secondly is, could that be targeted from a therapeutic standpoint, because it clearly distinguishes sort of the abnormal cancer cells from the normal healthy cells. Year 2 of the project has focused on differentiating stem cells to hematopoietic cells. So as part of this project, what I did is I created stem cells that are identical except for the presence or absence of one extra chromosome, chromosome 8, which is commonly gained in acute myeloid leukemia. And so now the second year of the project, I've really focused on taking those stem cells and then differentiating them into hematopoietic cells, because hematopoietic cells are the type of cells which leukemia arise in. What I've now been working on most recently is taking the hematopoietic cells, or the blood cells that we start with our stem cells, turn them into blood cells. Now I'm actually incorporating an acute myeloid leukemia oncogene, so MLLAF9, which is a gene that's mutated in AML and probably has a driving role. So I'm now incorporating that into our model system as well to try to study the effect of an acute myeloid leukemia oncogene in combination with 3 copies of chromosome 8 in blood cells. So in the future, what I think were really 2 directions we're sort of interested in taking this project. The first is to really, now that we have developed this system, to really understand what the interaction is between extra copy of chromosome 8 and what the role of it is in leukemia. And then the second part of the project would be to really take forward this idea of, can we target cells therapeutically based on extra copies of a single chromosome? And we've also been doing work on sort of trying to identify genes that might be important for the biology of cells that have 3 copies of chromosome 8. And then ultimately the idea would be to then try to target those genes and specifically kill cells that have 3 copies of chromosome 8, but not 2. I think there are a number of reasons why being a CureSearch Young Investigator has been really important. First of all, it's allowed me to actually complete this research. And obviously there's still a lot to do at this point, but I think we've got a really interesting system. We have new gene targets we're interested in following up on. And I think I couldn't have done that without the support from CureSearch. Secondly, I think it was also really helpful for my career development as well. I recently took a new job, and I think that I probably would not have gotten that job without having had support from CureSearch.